The overall goal of this procedure is to bring the 3DNA suite of programs to laboratory scientists and others to investigate DNA and RNA spatial organization with state-of-the-art computational tools. This is accomplished by first downloading the software from the 3DNA forum and installing it on a local computer. The second step is to analyze a representative DNA structure at the level of the constituent base pair steps. Next, the same DNA structure is modified at the base pair step level and is then rebuilt using the newly defined base pair step parameters. The subsequent step is the analysis of a large ensemble of related structures. The final step is the alignment of a set of related DNA structures on a common reference frame in order to visualize global distortions of the structures. Ultimately, the 3DNA tools are used to examine how individual base pair steps, including those bound to drugs and proteins, contribute to the overall folding of DNA and to identify the sites and directions of sizable global distortions.
The main advantage of this technique over existing methods is that the software is robust and widely applicable to a variety of different nucleic acid-containing structures. The method can help answer key questions in biochemistry and biophysics, such as how genomes are organized in 3 dimensions and what kind of deformations DNA can adopt.
To begin installation of the software package, connect to the 3DNA website at x3dna.org and click the link to the